Hello, my name is Saad, and today we're going to be looking at the bacterial preferences in C. elegans and C.Inopinata. So basically, in this experiment, C. elegans and C.Inopinata are the model organisms. C. elegans are found in rotting plants all over the world, whereas C.Inopinata are only found in figs prominently in Japan. So we're basically going to be observing the two nematodes and their behavior when it comes to two different types of bacteria, and that's basically the basis of this experiment. This is because C. elegans are lab adapted and C.Inopinata are not. So basically the question that arises is, do C.Inopinata and C. elegans have a preference over different types of bacteria? Part 1. So basically the methods, there's two parts to it. One was just identifying the bacteria. So this was done by a polymerase chain reaction, and it was basically amplifying the microbial 16S ribosomal DNA. This product was then sequenced using Sanger sequencing, and then the sequences were blasted on a database of other bacteria. So basically, after we got the sequence trace over here, we were able to put it into a program and identify the bacteria. The second part was actually observing the behavior. So the nematodes were placed on a petri dish, and all petri dishes were marked like this figure over here. Basically, the X represents where the worms were injected, and then each circle was either put with bacteria or medium, and these nematodes were then counted after 2 hours of being placed on the X to see what their preferences were. Now, during this experiment, or after this experiment, a Chemotaxis Index was also measured, and this was done through numbers of worms in spot A subtracted by the number of worms in spot B, which were then divided by the total number of worms in spots A and B. So using that Chemotaxis Index, we were able to get some results. Looking at this area over here, we can see and make the conclusion that bacteria was preferred over no bacteria. And looking over here, we can see that there was a very good distribution between the two bacteria strains, so there wasn't really much of a preference. So some key takeaways we can take are that the bacteria being identified. So strain 1 was Pseudomonas sp from the BLAST program that we got, and then strain 2 was Pantoea. The worms preferred bacteria over no bacteria, which was seen here, and then no bacteria was really preferred over the other, and that was seen between these two and all of these over here. And then the Chemotaxis Index was obviously evenly distributed, so we can see from 0 over here, and between 0 and +1 and -1 that they're pretty even. And so it basically indicated that there's not that much preference over bacteria. And some future experiments we could do is utilize certain amounts of bacteria or different concentration of bacteria to see if a higher concentration versus a lower concentration is preferred. Use of different types of bacteria. So basically, what we took away was that there was no preference, but what if we changed the type of bacteria and see if one was preferred over the other? And then, of course, male versus female, and to see if they have any preferences over bacteria.